Okay. So far, we've gone through the role of ATP in energy metabolism, and we've talked about our typical form of metabolism, which is glucose metabolism. So we talked about glycolysis, we had a little linking step involving pyruvate, we talked about the Krebs cycle, and we talked about oxidative phosphorylation using the electron transport chain. So now we're going to talk about the fact that the body is able to get energy from other things. So we probably all know that energy is partially stored in fats in the body, long-term energy storage. That's one of the purposes of fats in our body. And we can also break down proteins, and we can also break down glycogen, which is a storage molecule. So we want to think a little bit about how that works, how the body breaks those things down, and especially how the body gets to store energy for later, because we're not constantly eating, even if it is quarantine. Okay, so we're going to look at carbohydrates, lipids, and proteins, how they're stored and how they're used. This is the pretty basic pathway. So this is kind of the bird's eye view of the whole process. So here we can see our proteins. So that's one of the things we're thinking about, how it gets broken down. We can see glycogen, which is a storage carbohydrate molecule. So that covers our carbohydrates, carbohydrates that are not glucose. And here we see triglycerides. So triglycerides covers our fats. Love to get this bar out of the way. Okay. So we can see that these three molecules, or these three types of molecule, all feed into some pretty familiar pathways. So we can see glycolysis here. So that was our first part of glucose metabolism, of cellular respiration. We can see that glycogen is going to feed pretty neatly into this process. We can see that parts of our triglycerides also feed directly or partially into glycolysis. We can see that proteins, by contrast, get fed here somewhere around our Krebs cycle step. So they utilize these same pathways, but they're going to start a little farther into the process. So what you need to know here is not actually exactly where we're feeding into our cellular respiration pathways, but just understand that we're using a lot of those same reactions that we covered during glucose metabolism. We're just going to start at a different place, potentially, depending on the molecule we're talking about. So this is how we're going to get ATP from each of these other molecules. So we're going to feed these components into different parts of our cellular respiration pathway. And then the ATP comes from just the same steps as before. So from the Krebs cycle, from oxidative phosphorylation, or from glycolysis if we hop into these pathways early enough. Okay. One of the most important things to understand when we're talking about metabolism that isn't glucose specifically is glycogen metabolism. This is because glycogen is one of the first places your body goes to get energy when you run out of glucose. So this is a storage molecule, but it's kind of like medium-term storage. There's a lot of glycogen in your liver, specifically. We'll also see that it plays a role in the kidneys. We'll also see when we get to muscles that glycogen is going to come into play there as well. So mainly the liver, though, is storing glycogen, which is basically just a lot of glucose units chained together. So we chained together many monosaccharides, glucose, and we created this storage molecule, glycogen. Now, an important fact to know about glycogen is that as we start to break glycogen apart, as we start to take apart those glucose units, you don't immediately get just plain old glucose. What you actually get is glucose 6-phosphate as we start to break these apart. However, glucose 6-phosphate can't leave the cell, but glucose can. So before we can actually get our glucose to feed into glycolysis, if we're not staying in the liver or the kidney, we have to get out of the cell. So the liver and the kidney have this special enzyme, glucose 6-phosphatase, which converts glucose 6-phosphate to glucose, which is basically why we store glycogen in the liver and the kidney, is because in the liver and the kidney, we're able to break this molecule down, the glucose 6-phosphate, break it down to glucose so it can then exit the cell, enter the bloodstream, and get around to the rest of our body. So another thing that's happening in the liver is the synthesis of new glucose. So this is special. You don't actually have to start with a glycogen molecule, or you don't have to eat pure glucose. You can make glucose from other things. So in the liver, we're going to use glycerol, lactate, and amino acids. And we're going to be able to break them down, reform them into glucose molecules. This is called gluconeogenesis. So glucose, neo, new, genesis, creating something. So gluconeogenesis is how we create new glucose, mainly in the liver. So we don't do this by just reversing glycolysis, although that might be an easy thing to think of logically, right? Like, well, we end up with pyruvate at the end of glycolysis, so we could just run backwards and get glucose from pyruvate, which is partially true, but we're going to involve some other enzymes because some of our enzymes were specific to running glycolysis in a specific direction with specific components. So we're going to see that we can take glucose 6-phosphate, so our molecule that we saw from when we were breaking down glycogen, and we can use glucose 6-phosphatase to convert that to glucose. So that's what we saw just on the previous slide, right? So this was our glycogen metabolism, was turning that glucose 6-phosphate from the glycogen into glucose using glucose 6-phosphate. And we'll see we want to use an enzyme called hexokinase in order to get from glucose to glucose 6-phosphate, which we can then link together into, for example, a glycogen molecule. So if we want to look at the pathways of both breaking down glycogen and storing things in glycogen, we have a diagram kind of like this. So I think this figure is in your textbook. I'm going to explain to you exactly what's going on here. Okay, so this is showing us our glycogen starting at the top. So when we want to break down glycogen lysis. So lysis is always breaking something down. So glycogen gets broken down into glucose 6-phosphate. So one thing we can do is move that into the pathway and end up with pyruvate. So that's if we're like staying in the liver. We can also use glucose 6-phosphatase to turn that glucose 6-phosphate in the liver into glucose, which can then enter the bloodstream and be used by any cell in the same way it would use any other molecule of glucose. So we could send that glucose to your brain, break it down to provide energy to the brain. But another thing that we're going to want to be able to do is to store molecules as glycogen. So what we can also see in this pathway is that, say we had a buildup of pyruvate. I'm trying not to click down at the bottom there because that keeps pulling that up. But say we had a breakdown, a bunch of pyruvate in the cell. So we were breaking down a lot of glucose in our normal cellular respiration pathway. We're running a lot of glycolysis. We're also running the Krebs cycle and the electron transport chain, so therefore oxidative phosphorylation. But we have so much glucose coming in that we have really a lot of pyruvate. So one thing we would do at this point when we have an excess of glucose in the body is that we can convert the pyruvate into glucose 6-phosphate. So that's something that we can run backwards. So we can either convert glucose 6-phosphate into pyruvate or the reverse. So we'd convert pyruvate into glucose 6-phosphate. And then we can convert that glucose 6-phosphate into glucose. So that's one thing that we could do there if we have a buildup of the pyruvate. Or if we have just a buildup of the glucose right at the beginning of the process, we can use hexokinase, convert glucose to glucose 6-phosphate, and then we'd continue up this pathway. So we'd start with a bunch of glucose, use hexokinase to make glucose 6-phosphate, and then we'd link together those glucose 6-phosphate molecules into glycogen. So here they've shortened the term gluconeogenesis into glycogenesis, but it's a similar process. So we'd make glycogen there. So we're making glycogen. So the specific difference here is that gluconeogenesis is the part where we're making glucose, and glycogenesis is where we're taking the glucose and putting it into a glycogen molecule. So this is our storage, our glycogenesis. Okay. So that's a sort of medium-term kind of storage method. But if we want to have long-term energy storage, so on the order of, right, days, months, years, we're going to want to use a more efficient molecule to store our energy. Most energy is actually stored as triglycerides rather than glycogen. There's only a certain amount of glycogen your body can hold because it has to be in the liver and the kidney. They're only so big. But triglycerides can be stored all over your body. Most of them are stored in adipocytes, which are fat cells. So as we remember from going over lipids, our triglycerides are 3 fatty acid chains bound together by a glycerol backbone. And what's cool about these triglycerides and lipids generally is that they have a lot more energy per weight than carbs or proteins, which is to say that we can really pack a lot of energy into the bonds in our lipids, and we can store more energy therefore. It's a really efficient method of storage. And the reason for that is the way that fat metabolism works. So when we're talking about fat metabolism, here we're thinking really about a triglyceride. So our triglyceride has the 2 basic parts, the glycerol backbone and the 3 fatty acids. So first, we have a class of molecules called lipases. So lip for lipid, aces tells you it's an enzyme. We have lipases that break down a triglyceride to glycerol and those 3 fatty acids. So now all those parts are separate. So we basically have like 4 new kind of little molecules floating around. The glycerol can then get broken down on its own. So it can get broken down to dehydroxoacetone phosphate, which enters glycolysis. So that's this part that we see right here. So the glycerol goes into glycolysis, basically, and then it'll make us 20 molecules of ATP. The fatty acids undergo something special called beta oxidation, which basically works on just 2 carbons at a time. So we have that whole big fatty acid chain that was right. We had a backbone that was carbon, 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 and then we had hydrogens coming off each of them. The cool thing about beta oxidation, the like punchline of all of this, is that we can get 17 molecules of ATP for each 2 carbons in that fatty acid chain. And when we looked at the triglycerides, you would notice that those fatty acids, they're not just 2 carbons long, right? Like, could totally be 20 carbons long. And this is why we can hold so much energy in lipids as opposed to carbohydrates or proteins, is because it only takes 2 of those carbons from that fatty acid chain to get us 17 molecules of ATP. So technically the steps for this specifically is that each of those 2 carbons get converted into acetyl-CoA, NADH, one of our electron carriers, FADH, our other electron carrier. And then the acetyl-CoA can go into the Krebs cycle. The NADH and the FADH go to the electron transport chain. So that part is just like our normal aerobic cellular respiration. So just like when we were breaking down glucose, but we only had to start with 2 carbons. We only had to start with this 2 carbon little chunk of the fatty acid in order to get most of the benefit of oxidative phosphorylation. So we didn't feed in as many molecules of NADH and FADH, so we get a little less than we usually would, but it's still really efficient. Okay, so finally, protein metabolism. Protein metabolism works starting with something called proteolysis, which starts out by just breaking down our big protein or our polypeptide chain into its constituent amino acids. So amino acids, where our monomers are building blocks of the protein, proteolysis breaks them all apart. After proteolysis, so once we just have those amino acids, we have something called deamination. So deamination removes the amino group from the amino acid, leaving us with a keto acid and ammonia. Now, keto acids are what is going to feed into our cellular respiration pathways. So our keto acids can either be pyruvate or acetyl-CoA. Those are keto acids that we get through this process of proteolysis and deamination. And our keto acids, since they are pyruvate or acetyl-CoA, it can be those, they'd feed in directly right here, so they'd go directly into the Krebs cycle. So this is where the proteins would kind of jump into our process of metabolism. Okay. So questions about these other types of metabolism, they're not a big emphasis of this course, but there's something that I'm always curious about when we start talking about how we break down glucose, because we eat a lot of stuff that's not glucose. So I thought we should talk about that in a little detail before we move forward.
I have kind of a vague question about how you were saying that we can get energy from more than just oxygen or like consuming things. Do you think that that's how small, like other organisms that could go a really long time without oxygen or without eating might get some of their energy?
I'm not sure I understand your question, so I'm going to repeat what I think your question was.
Oh,
you can tell me if I'm answering you. So you're just asking about how small organisms in like low oxygen environments get energy, because that's kind of a separate question to this breaking down of
yeah, it's kind of different from like human anatomy and phys. It was just something that popped into my head when we were thinking about like how all other ways that, you know, our cells can function.
Yeah, so they basically do a specialized kind of process of fermentation. So when we talked about muscle cells and like when you're exercising, when we talked about creating lactic acid, sometimes we call that lactic acid fermentation when we break down glucose and we just do glycolysis and then take that pyruvate to get us our NAD plus again to keep going. Organisms like yeasts have their own process that's similar to that, but instead of making lactic acid, they make ethanol. So that's actually how like beer production works, is that that's a product of just glycolysis running over and over again. And glycolysis doesn't need oxygen. So that's how a lot of organisms do it, basically, if they're in low oxygen environments, whether that's permanently because that's where they live, or whether they just end up in a vat of beer where they've used all the oxygen already. They do fermentation, which is the general term for that conversion of pyruvate back into the, that conversion of pyruvate that allows us to regenerate the NAD that we need for glycolysis.
Oh, okay.
Does that answer your question?
Yeah.
Okay.
Mm-hmm.
Okay. If that's it on metabolism, we're going to move into membrane transport, as you might expect by this big slide that's up. I linked that in the slides pretty recently, so that is on Brightspace. This is moving into basically our final section before the exam, but it's a big chunky one. So go ahead and pull that up. We're going to start with something that should be pretty basic review. We're going to start by talking about the plasma membrane and its properties, but then we're going to move pretty quickly into talking about gradients, and especially something called the electrochemical driving force, which is going to be really important as we start to talk about the nervous system in our next unit. So it's really important that we understand how this stuff is working, but we're going to spend a couple days on it. So we're going to start out by talking about phospholipids and specifically how phospholipids compose the plasma membrane and what properties that gives the plasma membrane. And then we're going to go into chemical and electrical gradients, the electrochemical driving force, and we're going to be starting with that in order to help us determine whether we're going to have passive transport of substances across a membrane or active transport. So after we go through the details of the electrochemical driving force, we're going to be hopefully back in familiar territory for you guys and talk about active and passive transport. Okay. So starting out, we're going to look at the fluid mosaic model of the plasma membrane. I'm going to do a little drawing. Am I am. Okay. So when we're talking about the fluid mosaic model of the plasma membrane, fundamentally, we're just talking about the fact that a plasma membrane is not just phospholipids. It has other parts, and they sort of shift relative to each other. They're sort of fluid. They're able to move. So that's talking about what a plasma membrane looks like when you look at it, how it's behaving in some kind of 3D space. So I left a lot of room here so that we can draw a plasma membrane, and then we're going to get into each of the parts. But pretty basically, we're going to start with some phospholipids. So they have polar heads. They have nonpolar tails. So our polar heads are hydrophilic. Our nonpolar tails are hydrophobic. So they're going to face off because there's going to be water inside our cell and there's going to be water outside our cell. So I'm going to label this extracellular fluid, or ECF, over here. And we're going to have our intracellular fluid, or ICF, here on the inside of our cell. So we need something hydrophilic touching this water and touching that water. So that's going to be our polar heads of our phospholipids. And we'll have a big lineup of these. Drawing with my mouse is hard, so I'm not going to do that many. Hopefully your drawings are prettier than mine. But this is the basis of our plasma membrane. And this is what's going to lead to a lot of the properties of the plasma membrane. Because the fact that we have these polar heads on the outside, these hydrophilic heads, because they're near water, that's great. That's protecting these nonpolar tails from having to touch this water. But it also means that they're going to repel certain molecules. So they're going to repel some ions. They're going to attract them a little bit. We'll see how we have slight gatherings along the edge of the membrane. So this is going to affect how the outside of the cell interacts with the concentrations of different things in that fluid environment. But these fatty acid tails, so these tails, these nonpolar tails on the inside of this bilayer, form a really thick hydrophobic area. So anything that's attracted to these polar heads is basically going to get blocked by this hydrophobic nonpolar area. So things with charges may be attracted to the membrane, but they're not going to be able to get across without help because they can't cross through this hydrophobic region of the plasma membrane. So that's why it's really important to understand the structure of a phospholipid, because it's going to control what can go through directly across the membrane and what's going to need assistance. Okay. We're also going to see that our mosaic model here of our plasma membrane is going to include cholesterol. Okay, so we have our cholesterol molecules, so that's another type of lipid. It'll be in an image later. So we saw that cholesterol is the basis for our steroids, which is a class of lipid, and it's going to give some structural properties to our plasma membrane. And then we're also going to have membrane proteins. So we'll see that these become really important when we're talking about active transport especially, but also passive transport. So membrane proteins cross the entire membrane. So we'd have more of our phospholipid bilayer over here. So they actually have both polar parts on the outside and nonpolar parts on the inside that can touch this nonpolar region of our phospholipid bilayer. But because proteins are so big, they're going to basically be able to allow things to pass through the center of them without getting blocked by nonpolar layers. And that's because a protein is so large, you could have potentially a nonpolar part over here, but have, say, a channel on the inside of our protein that's protected and allows things to flow through. And then we'll also have membrane carbohydrates, which are kind of easy to forget about because our carbohydrates in the membrane are just going to be like little chains coming off of different things. So we'll have little carbohydrate chains coming off lipids and coming off the outside of proteins sometimes. And these are important for cells to recognize each other and sometimes to bind to each other. Okay. So looking through a slightly better drawing of these things, we have our phospholipids. So our phospholipids are the basic structure of the membrane. So we can see them on this drawing in blue here. Okay. And they're the barrier to the passage of water-soluble substances between the ICF and ECF, which is basically just saying, so something that's water-soluble is something that can dissolve in water. And water itself has polar properties. So because water has polar properties, one of the coolest things it does is form hydrogen bonds. You might have talked about that in your other classes. But one of the other things that's true about water because of this polarity is that ionic bonds dissolve in water. So we end up with ions, which are basically little charged particles. So things that are water soluble, basically we're talking about ions. So they're going to be little pieces of things with little charges on them. So this phospholipid barrier is going to prevent these little charged particles from flowing directly across the membrane because they have this big hydrophobic layer in the center. So here we see our fatty acid tails. It's labeled that they're nonpolar. So that's blocking anything that's water soluble, probably has a charge, so it can't pass through that center. Phospholipids also provide fluidity to the membrane. So even though this looks like a big sheet of phospholipids, they're not actually like covalently bonded to each other. They can jiggle up and down. So the fact that they can do that provides some fluidity to the membrane. So we'll see that lipid soluble things, so things that are non-polar, will sometimes actually be able to squeeze between phospholipids. And we'll get to some examples of that as we move forward. The phospholipids are impermeable to water, right? Our whole point of arranging in this bilayer is to block the water on the outside of the cell and the extracellular fluid and from the inside of the cell, so the intracellular fluid, the cytosol. So they're not going to allow water to cross, except we're going to have a very special structure called an aquaporin in basically every one of our membranes. We'll get to some exceptions when we get to kidneys. But we're going to have this special membrane protein called an aquaporin. There are lots of them. So fundamentally, when we think about cells, we think of them as being, as allowing water through because of this presence of aquaporins. We're going to talk about membrane proteins in a second, but since we just drew some out, so like imagine that this, for example, is an aquaporin. So that's actually how water would get across our cell membrane. Okay, so looking at the cholesterol, so here we see in yellow, we have this little cholesterol molecule. We can recognize it's a steroid due to these sort of 4 rings pressed against each other. It also adds fluidity to the cell membrane. So one of the things that could happen if you had a plasma membrane that's just phospholipids is that even though they're not covalently bonded to each other, they could kind of settle into an almost crystallized state in that non-polar hydrophobic layer on the inside of the membrane. And so the cholesterol basically just kind of breaks that up. So we have a cholesterol molecule here and there. So it messes with the arrangement of these tails so they don't end up locking together. Okay. This decreases the permeability of the membrane to water. Just a fact about cholesterol. Not very permeable to water. The presence of cholesterol is not going to allow a lot of water through. So those are our 2 types of lipids that we have in the plasma membrane, our phospholipid and our cholesterol, both lipids. Now we're moving on to proteins, and we'll see that we have proteins basically of two types. We're going to see integral membrane proteins. We'll see peripheral membrane proteins. And these are basically just labeled based on how much of the membrane they go through. Okay, so if you wanted to just pull out a protein, I don't know really why you would want to do this, you'd have to disrupt the membrane. The integral membrane proteins that go all the way through the cell membrane do have their own amphipathic properties. So amphipathic is a word we associated before with phospholipids. Amphipathic means it has a hydrophilic part and a hydrophobic part. So our integral membrane proteins that go all the way through the cell membrane are just like the phospholipids. They have a part that's hydrophilic and they have a part that's hydrophobic. So these proteins go all the way across, partially because they can nest in here with both the polar phosphate heads of the phospholipids and with these non-polar chains in the middle. So some of our proteins are transmembrane proteins. These are our channels and our carrier proteins. We'll get into those more specifically as we start to talk about active transport. We'll also see that some proteins are located on just one side of the membrane. And we'll have our peripheral membrane proteins here. So enzymes and receptors may not actually go all the way through the cell membrane. They may be on one side and just have one specific job, either involving the inside of the cell. So sometimes we'll have enzymes that are actually stuck in the cell membrane on one side of the cell. And so they'll catalyze chemical reactions that way, like from the membrane, but not move around on the inside of the cell. We'll also have various receptors. So we'd expect receptors mostly to be on the outside of the cell, although there are exceptions. So we'd have receptors on some part of the cell receiving signals from other cells. So those would also count as membrane proteins. Okay. So peripheral membrane proteins, so ones that are really kind of just on one side of the membrane, are easier to dissociate from the cell membrane because they're just on one side. They don't go all the way through. They're actually not even inserted in the cell membrane. So we can see here that we have a membrane protein that's kind of sitting on top of these phosphate heads here, these polar heads of the phospholipid. So it would be easy to rip off this peripheral membrane protein. Most of them are on the cytosolic side of the membrane. And if you've talked in detail about the structure of the cytoskeleton, a lot of these peripheral membrane proteins are basically part of the cytoskeleton. So we can see here we have some cytoskeleton linking in towards this peripheral membrane protein. Okay. So finally, we have our membrane carbohydrates. So we have little carbohydrate chains either bound to proteins or to lipids. So a glycoprotein is a protein with a carbohydrate chain bound on it. So that's one type of membrane carbohydrate. It's like a partnership between a protein and a carbohydrate, glycoprotein. Glycolipid is the same thing, but we're binding on to some type of lipid, probably a phospholipid, right? So here we have a glycolipid. It's sitting on this polar head of our phospholipid. So the whole structure together, the carbohydrate part and the phospholipid part, we'd call a glycolipid. So glyco, remember, is referring to basically glucose. So that's how you can kind of remember that that's the name for a carbohydrate in the membrane. So these also have some cell functions. So one of the most important ones is cell recognition. They also function to create something in a called a glycocalyx in some cells. We don't talk about this really much in human anatomy, but it is important for some other organisms, some microbes, basically forming almost another layer outside the cell membrane. Okay. Occasionally, you'll see something analogous to that in epithelial tissues. Okay. So those are the pieces of the membrane. The reason we really care about membranes, other than, they are in the human body, and this is a course on the human body, the reason we really care about the membrane is that these membranes are going to allow for transport. So they're going to allow things to cross that membrane. And they're also going to block some things out. So our membranes are selective in what can go across, which means that we can maintain different environments inside the cell versus outside the cell. So we saw that that's kind of really important for homeostasis, right? We want to be able to control environments. We want to be able to regulate processes. So it's important that we can maintain the inside of our cell, potentially different from the outside of the cell, which can be more affected by external conditions. Okay. So when we think about the membrane, we really want to focus on the fact that they're separating stuff. So a membrane separates the inside of the cell from the outside of the cell. More scientifically, we say that the membrane separates the intracellular fluid from the extracellular fluid. So that calls back to when we were talking about different fluid compartments, right? Okay. So we're going to allow some material to get exchanged between the intracellular fluid and the extracellular fluid, but not all of it. We're going to control very specifically what is allowed to cross. It is important that stuff can cross because the inside of the cell, we're going to need oxygen to run cellular respiration, right? We need to make that ATP so our cell can do stuff. We're going to need other nutrients for other processes going on in the cell. We're also going to need to get rid of waste products. So we're producing waste in the form of CO2, for example, while we were running cellular respiration. We want to get rid of that. We don't want it to build up inside a cell. So we're going to need it to cross out of the cell into the extracellular fluid. So specifically, eventually into our plasma, into our blood, from where we can get rid of it. So we call this fact that we are choosy about what can cross the membranes. And when I say choosy, I mean we're able to choose based on like what membrane proteins we insert in that membrane. We're selectively permeable in that we allow some molecules to pass through, but not all of them. So permeable, when we say a membrane is permeable to something, we mean that that thing can cross. So if I say the membrane is permeable to oxygen, I'm saying oxygen can go across that membrane. And specifically, oxygen is one of the things that can cross directly between those phospholipids. So we wouldn't need a protein for oxygen. But our membrane is also selective. And so when we say it's selective, what we really mean is restrictive. So we mean that it's blocking stuff. So often ions. So when we're thinking about specific molecules and we're trying to think about whether the selectively permeable membrane is going to allow them to cross, we want to think about their polarity. So non-polar molecules, especially small ones, will be easily transported across the membrane. So some examples of non-polar molecules are oxygen, CO2, fatty acids. None of them are polar, so they don't get blocked by the hydrophobic section in the middle of the bilayer, so they can go directly across. So we'd think of these as lipid-soluble molecules. But ions and polar molecules have either actual charges in the case of ions, or like little pieces of charges in the case of polar molecules. And they're not going to be able to cross the membrane on their own. If we want them to cross, we're going to have to transport them, so we're going to have to use a protein. So examples of ions and polar molecules that we'll see interacting with our cells are glucose, proteins, and we'll see a lot of ions, specifically sodium. We're going to talk about sodium like all the time in this course. Okay. So in order to understand what is crossing into cells, what is crossing out of cells, when and how, we need to understand gradients. So we're going to talk about chemical gradients, electrochemical gradients, and electrochemical gradients. So we're going to basically do two component pieces and then put them together to understand what can passively move across a membrane, so without adding in energy, and what needs to be actively transported across a membrane, so where you're going to have to plug in some ATP in order to push something across. So when we say we're talking about a gradient, A gradient comes from the word for degree, which is a way you can measure how sharp a slope is. So we're basically talking about a hill. So when we talk about passive transport, we're talking about like this orange ball rolling down this hill. This happens spontaneously, right? Like stuff just does roll down a hill. Stuff rolls down a gradient also. So when we're thinking about this hill, we want to think, for example, as the top of the hill being a high concentration of something. And here we have a low concentration of something. So we're going to go down gradients from high to low. So whenever we're thinking about specific examples, you want to think about where our high is and where our low is, because passive transport, our automatic sort of spread of molecules, is going to lead things to go down this gradient. Active transport is the reverse. So again, I'm using the example of chemical gradients, but we'll also look at our electro gradients and our electrochemical gradients. So if we're talking about active transport, we're trying to go up the hill. We're going from a low concentration to an area of high concentration. Going up a hill takes energy. It's hard. We're going to require ATP. It's going to be active transport. It's not spontaneous. Okay. So everything we're talking about as we start talking about gradients and driving forces is really to figure out whether transport is going to be passive and spontaneous or active and non-spontaneous. Okay. So when we're thinking about things moving across the membrane, we're going to be thinking about both the chemical force on whatever specific molecule we're tracking is. We're also going to think about the electrical force on that molecule, or really ion. We're mainly going to be talking about ions here. Okay, so any given ion is going to have a chemical gradient. So any given ion has an area of low concentration and an area of high concentration, if there's a difference in concentration. But any given ion is also going to have potentially an area that attracts it and an area that repels it. So that's where we're going to get this electrical force from. And if we put the two together, we're going to call it the electrochemical force. So the chemical gradient is going to be pushing that ion in some direction. Specifically, the chemical gradient is going to want that ion to move towards the area of low concentration, so down that hill. The electrical forces are going to want to move that ion away from an area of similar charges and towards an area of opposite charges, because opposite charges attract. So we're going to have to add these two forces together in order to look at the electrochemical force to figure out whether movement is passive or active, because our molecule isn't going to get like pushed by the chemical force for the first second and then by the electrical force for the second. Both of these forces are going to be acting at the same time. So in order to predict which way our ion is going to move, we need to add them together. We're going to talk about the chemical driving force first, which we'll see expressed as CDF in some future diagrams and slides. So our chemical driving force, or CDF, is really just talking about the concentration gradient. So our concentration gradient means we have a difference in particle concentration between the inside of the cell and the outside of the cell. So we have more of this particle in one place and less somewhere else. We're going to basically be pushing particles from areas of higher concentration to lower concentration. So they're going to be moving downhill or down their gradient from the high point to the low point. Okay. So if we're looking at this diagram A, say we're looking at the cell, we have this red particle. We have a high concentration of this red particle inside the cell, and we have a low concentration outside the cell. So you can see how it's packed in there inside the cell, and it's really spread out outside the cell. So our chemical driving force in this diagram would move towards the outside of the cell. So the driving force is pushing any given red particle out of the cell in order to reach equilibrium across the membrane, in order to even out the spacing between these molecules. Okay. So here we can see the reverse. So if we have a high concentration outside the cell, a low concentration inside the cell, this packing of this solute, of this molecule, of this particle, of this ion, whatever it happens to be, the chemical driving force would push that inward, because again, for our chemical driving force, this molecule really just wants to spread out, just wants to reach equilibrium, just wants to have even concentration anywhere. So our chemical driving force, in order to make that happen, would push this particle into the cell. So our direction of the chemical driving force is always going to go from higher to lower concentration, which we refer to as down the chemical gradient. Okay. This is the last slide we're going to do before we break and pick up on Friday, but this is going to really prepare us to understand the electrical driving force. So we want to get some very basic terms down before we start talking about the electrical driving force. When we're talking about the electrical driving force, we're not talking about any old molecule. Really, we're talking about things with charges. So when we're talking about oxygen, for example, nonpolar, not charged, not an ion, we wouldn't have to worry about the electrical driving force. For oxygen, we'd really just be looking at the chemical driving force. But for an ion, say a sodium ion, right, Na+, it's charged. It has a charge on it, which means that we have to take those charges into account. So a cation has a little plus in there, right? This T is like a plus. A particle with a positive charge is called a cation. A particle with a negative charge, so the opposite, so NaCl is plain old table salt, so sodium has a positive charge, Cl, our chlorine, has a negative charge, so that would be our anion, the chlorine, a chloride ion, I should say. Okay, so our ions have either a positive charge or a negative charge. And our intracellular fluid versus our extracellular fluid is not always going to have even amounts of cations and anions. So we're also often going to see a charge separation where we have, for example, a lot of anions inside the cell and therefore a negative charge inside the cell. And in fact, that's typically what we see. So usually in a cell at rest, the inside of the cell is more negative than the outside, and the most common amount of negative we have inside is measured as negative 70 millivolts. So you don't have to worry about that specific number for right now, but when we talk about neurons, we're going to be talking a lot about changes to that voltage, so getting it out of the way right now. Okay, so we refer to this separation of charges as the membrane potential. So this separation in charges basically is a force. So the fact that you have negative in one place, you have positive someplace else, basically means that wherever you have a negative charge, that's going to repel negative ions. Wherever you have a positive charge, that's going to attract negative ions, and the reverse. So wherever you have the positive charge, that's going to repel positive ions, so we're repelling the cations, and then wherever we have our negative charge, it's going to attract cations. So this unequal distribution of the anions and cations causes a force that we're going to need to take into account when we're trying to figure out like which direction an ion is going to move. Okay. The strength of that force is measured in volts, really in millivolts because it's quite small. Okay. And the last thing to know about it is that it has a polarity. So we're going to see either negative numbers or we're going to see positive numbers. We're also going to basically reference the normal resting membrane potential, we're going to be referencing the internal cellular fluid. And we'll talk about what that means when we get together again on Friday and talk about the electrical driving force and how it leads to the electrochemical driving force on any given ion.